From the subtle blink of an eye to the explosive power of a sprint, our muscles are, well, truly one of the marvels of the human body. They seem simple, right? But underneath, there's this incredibly complex, microscopic world just humming away.
Absolutely. It's fascinating stuff.
So today on the deep dive, we're pulling back that curtain, our mission, to unpack the dense yet absolutely vital information from Walter F. Boron and Emile L. Boulpaep's Medical Physiology, specifically Chapter 9. We're talking the cellular physiology of skeletal, cardiac, and smooth muscle.
And the goal is to make it clear, engaging, accessible.
Exactly. We'll break down these complex ideas from the ground up. Think of this as your shortcut to being really well-informed on this critical topic without feeling like you're drowning in detail. By the end of this, you'll have a solid foundation how these different muscle types work, how they're controlled, and, crucially, why all these tiny details matter for actual clinical understanding. Diagnostics, pathology, treatment.
Right. Because what's fascinating here is just how fundamental this knowledge really is. Understanding muscle physiology isn't just academic, it's the basis for diagnosing, understanding, and treating so many conditions.
It really underpins so much of medicine.
It does. And while these muscle types look and act very differently, they all share a basic purpose, and, as we'll get into, this one critical common trigger for contraction. It's a beautiful system of shared principles, but unique specializations.
Okay, let's unpack this, starting with that big picture.
The three muscle musketeers. An overview.
So at its core, what does muscle do? Its main job is generating force or movement. That's it. But it does this in response to some kind of physiological signal.
And our bodies have evolved three distinct types to handle different jobs.
First up, skeletal muscle. This is the one you think about most, the muscles attached to your bones that you consciously control. For moving, lifting things, even breathing, thanks to the diaphragm.
Right, the voluntary stuff. Then there's cardiac muscle, found only in the heart. Its whole purpose is to be a biological pump, pushing blood around your body constantly.
Non-stop, hopefully.
Hopefully. And finally, smooth muscle. This is kind of the unsung hero working behind the scenes. It controls things like your digestive tract, your bladder, blood vessel diameter, airways, all sorts of internal plumbing.
So different jobs, and as you said, different personalities. Skeletal muscle needs to contract strongly, maybe hold that contraction. Cardiac muscle needs those brief, rhythmic contractions for life.
And smooth muscle is the master of sustained, often low-level contractions without getting tired. Think blood pressure regulation. But despite all these differences in speed, duration, how they use energy, how easily they fatigue.
There's that common thread you mentioned.
Exactly. The ultimate trigger for contraction in all three types is a rise in the free calcium concentration inside the cell. That cytosolic calcium, Ca2 plus I. That's the universal go signal.
Skeletal muscle, the movers and shakers.
Okay, let's zoom in on skeletal muscle then, the voluntary kind. It's got this amazing hierarchical structure.
It really does. You start with the whole muscle, say your biceps. That's wrapped in connective tissue. Inside you have bundles called fascicles.
Okay.
Each fascicle contains many individual muscle fibers, which are the actual muscle cells, also called myofibers.
Got it. Cells within bundles within the whole muscle.
Exactly. And inside each muscle fiber, you have these long cylindrical structures called myofibrils. And these are made of repeating units end to end called sarcomeres. That's the fundamental contractile unit.
Sarcomeres. Okay, that's the key structural bit for contraction. So how does the brain's command get down to these sarcomeres to make them, shorten?
It starts with a nerve cell, a motor neuron. A single motor neuron will branch out and connect to several muscle fibers. That neuron plus all the fibers it controls, that's called a motor unit.
Ah, the motor unit. I've heard that term.
Right. So the signal comes down the motor neuron, reaches the connection point, the neuromuscular junction, and releases a chemical messenger, acetylcholine or ACH.
Okay, ACH crosses the gap.
It binds to receptors, specifically nicotinic receptors, on the muscle fiber's membrane, the sarcolemma. This causes a local electrical change, the end plate potential. If that's strong enough, it triggers a full-blown action potential, an electrical wave that spreads across the entire muscle fiber surface.
And this motor unit idea, the number of fibers per neuron, that varies, right? That's important.
Hugely important. It's called the innervation ratio. For really fine control, like moving your eyes, one neuron might only connect to, say, 3 muscle fibers. Tiny ratio, exquisite precision.
Makes sense.
But for generating large forces, like in your big leg muscles for jumping, one neuron might control hundreds, maybe even 1,000 fibers. Large ratio, lots of power. It's elegantly designed for function.
Incredible. So that action potential is racing across the surface. How does it get inside the fiber to the myofibrils?
Ah, this is excitation-contraction coupling, or EC coupling. The muscle fiber membrane isn't just flat. It has these deep invaginations like tunnels, called transverse tubules or T-tubules.
T-tubules, okay. Like plumbing running into the cell.
Exactly like plumbing. They dive deep into the fiber, running right alongside the myofibrils. This allows that surface action potential to penetrate rapidly into the cell's interior.
And what's down there waiting for the signal?
The T-tubule meets up with the sarcoplasmic reticulum, or SR, that's the cell's internal calcium storage tank. Specifically, the T-tubule snuggles up against two enlarged parts of the SR called terminal cisternae. This whole structure, one T-tubule and two adjacent SR cisternae, is called a triad.
The triad, okay. T-tubule plus SR calcium stores. How does the electrical signal jump the gap?
It's a really cool mechanism in skeletal muscle. On the T-tubule membrane, there are voltage-sensitive proteins called L-type carome-2 plus channels, also known as DHP receptors. When the action potential arrives, these channels sense the voltage change and physically change their shape.
They act like sensors.
Right. And here's the key. They are physically linked, almost like they're holding hands, with another set of channels on the SR membrane. These are the Ca2 plus release channels, or ryanodine receptors, RYR.
Ryanodine receptors on the SR.
Okay. So when the L-type channel changes shape due to the voltage, it literally pulls open the ryanodine receptor it's linked to. It's a direct physical electromechanical coupling.
Wow, so it's not necessarily calcium coming in that triggers it.
In mammalian skeletal muscle, calcium influx from outside through those L-type channels can happen and can contribute, but it's not required. The physical link is the primary trigger. This direct connection opens the ryanodine receptors and whoosh, calcium floods out of the SR into the cytoplasm surrounding the myofibrils.
And that calcium flood is what kicks off the contraction at the level of the sarcomere.
Precisely. Now we're at the molecular machinery.
Right, the sarcomere, which gives the muscle that striped or striated look. You mentioned thin and thick filaments.
Yes. The thin filaments are mainly made of a protein called actin, arranged like two intertwined strands of pearls. Attached to actin are regulatory proteins, tropomyosin, a long filament that lies in the groove of the actin strands, and troponin, a complex of 3 subunits. One of these subunits, troponin C, TNC, is the calcium sensor.
Okay, actin, tropomyosin, troponin C for calcium, and the thick filaments.
The thick filaments are bundles of a protein called myosin 2. Each myosin molecule has a long tail and 2 globular heads. These heads are crucial. They have a site to bind actin and a site to bind and hydrolyze ATP, the energy molecule.
Got it. So myosin heads want to grab onto actin. What stops them normally?
In a resting muscle, when calcium is low, that tropomyosin filament is physically positioned over the myosin binding sites on the actin. It's like covering the docking ports. Troponin helps hold it there.
So tropomyosin is the blocker. How does calcium remove it?
When calcium floods out of the SR, it binds specifically to those low affinity sites on troponin C. This binding causes troponin to change shape. And that shape change pulls the tropomyosin filament slightly, uncovering the myosin binding sites on the actin. The gate is now open. Myosin heads can bind to actin.
And that binding starts the cross-bridge cycle, the actual force generation.
Yes, the molecular dance. It's powered by ATP hydrolysis and involves the myosin heads repeatedly binding to actin, pulling, and then detaching. It's a cycle.
Can we walk through those steps?
Sure. Let's start with the myosin head attached to actin. Step 1, ATP binding. A fresh molecule of ATP binds to the myosin head. This binding causes the myosin head to detach from the actin filament.
Okay, detachment first. This explains rigor mortis, right? No ATP, no detachment.
Exactly. Muscles stay stiff. Step 2, ATP hydrolysis. The ATP bound to the myosin head is split into ADP and inorganic phosphate, pi. The energy released causes the myosin head to cock or pivot into a high energy position, sort of ready to spring.
Like pulling back a lever.
Kind of. Step 3, cross-bridge formation. The cocked myosin head now binds weakly to a new site further along the actin filament.
It gets reattached.
Step 4, release of pi and the power stroke. The inorganic phosphate pi is released. This release triggers the power stroke. The myosin head pivots forcefully back to its lower energy position, pulling the thin filament along with it towards the center of the sarcomere. This is the force-generating step.
That's the actual pull.
That's the pull. Step 5, ADP release. Finally, the ADP molecule is released from the myosin head. The head remains tightly bound to actin in this state, waiting for a new ATP molecule to arrive and start the cycle again by binding and causing detachment.
Wow. A constant cycle of bind, pull, detach, recock, bind again, fueled by ATP.
Millions of times per second in an active muscle, which means you need a lot of ATP. Cellular stores only last a couple of seconds of intense activity.
So how do we regenerate it so quickly?
Several ways. The fastest is using phosphocreatine. It can quickly donate its phosphate back to ADP to make ATP, good for maybe 10 seconds of burst activity.
Okay, the quick fix.
Then there's breaking down stored glycogen into glucose, which can feed into energy pathways. For longer-term, sustained activity, oxidative metabolism is key, using oxygen to efficiently generate lots of ATP from glucose or fats.
And if oxygen runs low?
Then anaerobic metabolism can take over for a bit, making ATP from glucose without oxygen, but it's less efficient and produces lactic acid. It can sustain activity for maybe a minute or so.
Right. So after the contraction, the nerve signal stops. How does the muscle relax? Calcium needs to go back where it came from, right?
Exactly. Relaxation requires lowering that cytosolic calcium concentration back down to resting levels. Some calcium gets pumped out of the cell entirely.
But most goes back into the SR.
Yes, the vast majority is actively pumped back into the sarcoplasmic reticulum by a pump called the SERCA pump. It uses ATP to move calcium against its concentration gradient back into storage.
So SERCA clears the calcium, troponin changes back, tropomyosin blocks the sites again, and the cross bridges release.
And the muscle relaxes. Inside the SR, proteins like calsequestrin bind up a lot of that calcium, which helps keep the free calcium concentration low inside the SR, allowing the SERCA pump to keep working efficiently.
Okay, makes sense. Now let's talk mechanics. How do we measure what the muscle actually does?
Good question. We often study muscle under controlled conditions. One is isometric contraction. Iso means same, metric means length. So the muscle contracts, generates tension, but its overall length doesn't change because the load is too heavy or fixed. Think pushing against a wall.
Okay, building tension, no movement.
The other is isotonic contraction. Tonic means tension or tone. Here, the tension stays relatively constant while the muscle does shorten because the force it generates is greater than the load. Think lifting a weight.
Right, movement occurs. You mentioned before that the force a muscle generates depends on its length.
Yes, the length-tension relationship. If you measure the maximum isometric force a muscle fiber can generate at different starting lengths, you find something interesting. If the muscle is too short or stretched too long, it can't generate much active force from the cross bridges.
Why is that?
At very short lengths, the actin filaments overlap too much or run into the Z-discs, interfering with cross-bridge formation. At very long lengths, the actin and myosin filaments are pulled so far apart that there's very little overlap for the myosin heads to grab onto.
So there's a sweet spot.
Exactly. There's an optimal length, L0, usually near the muscle's normal resting length, where the overlap is perfect, allowing the maximum number of cross bridges to form and generate the most force.
That makes perfect sense visually. What about the speed of contraction?
That's the load-velocity relationship. It's pretty intuitive. The heavier the load the muscle is trying to move, the slower it shortens.
Lift a feather fast, lift a bowling ball slowly.
Precisely. At zero load, the muscle shortens at its maximum possible velocity, Vmax, which depends on how fast the myosin heads can cycle. As the load increases, the velocity decreases. Until you reach a load, the muscle can no longer lift, isometric condition, where velocity is zero.
Why does heavier load slow it down?
Because more cross bridges need to be engaged just holding the load, leaving fewer available to contribute to the actual shortening movement at any given moment. Think of it like having fewer hands free to pull the rope when you're also busy holding it steady against a heavy weight.
Okay, that clicks. So in our bodies, we rarely just activate one fiber. How do we control the overall force of a whole muscle, like lifting something light versus something heavy?
Two main ways. First, temporal summation or frequency summation. We talked about this a bit. Because a single muscle twitch lasts longer than the action potential that caused it, if you send action potentials close enough together.
Before the first twitch fully relaxes.
Right. The twitches start to summate, adding up. Increase the frequency of action potentials enough, and the twitches fuse together into a smooth, sustained maximal contraction called tetanus. This is due to calcium levels staying high in the cytoplasm.
So faster nerve firing equals stronger, sustained contraction in that fiber. What's the other way?
Multiple fiber summation, or spatial summation. This is about recruiting more motor units. Your brain controls how many motor units are active in a muscle at any given time. For a weak contraction, maybe only a few small motor units are activated.
And for a strong contraction?
You recruit more and more motor units, and typically larger ones get added as the demand for force increases. This is the size principle. Smaller, more easily excited motor units are recruited first, then progressively larger ones.
That seems efficient.
It is. And the brain usually activates these motor units asynchronously, meaning they fire slightly out of sync with each other. This helps smooth out the overall muscle contraction, preventing jerky movements and allowing for sustained force without fatiguing all fibers at once.
Very clever design. Now, you mentioned things can go wrong. Let's talk about that clinical connection. Malignant hyperthermia. Sounds serious.
It is very serious. MH is a genetic disorder, maybe affecting 1 in 10,000 people, maybe fewer. It's triggered by certain inhaled anesthetics, like halothane or the muscle relaxant succinylcholine.
What happens when it's triggered?
It's traumatic and dangerous. The person develops rapid breathing, low oxygen, high CO2, a very fast heart rate, muscle rigidity, and, critically, a rapidly rising body temperature, like a degree Celsius every 5 minutes, plus wild blood pressure swings.
A degree every 5 minutes. That's incredibly fast. What's causing that heat?
It's uncontrolled, sustained muscle contraction. The muscles are basically locked in a state of extreme activity. This burns through ATP at an incredible rate. All that metabolic activity generates enormous amounts of heat. It also leads to acidosis and muscle breakdown.
And the genetic link is often, too, that calcium channel.
Exactly. Often it's mutations in the RYR1 gene, which codes for the ryanodine receptor on the SR. These mutations make the channel leaky or hypersensitive, so it stays open longer or opens too easily, leading to uncontrolled calcium release into the cytoplasm. The calcium floodgates are stuck open.
Yikes.
Yeah.
How is it treated? It sounds like an emergency.
It absolutely is. The key is immediate administration of a drug called dantrolene. Dantrolene specifically works to block excitation-contraction coupling, interrupting that runaway muscle contraction.
So it directly targets the problem mechanism.
Yes. Alongside dantrolene, you have to stop the triggering anesthetic immediately, aggressively cool the patient, and provide intensive supportive care, IV fluids, managing acidosis, supporting breathing. Even with prompt treatment, it can still be fatal, which highlights why screening family members is so important if a case is identified.
Cardiac muscle, the unstoppable pump.
Okay, shifting gears now from skeletal muscle to the heart cardiac muscle. Structurally different, right?
Very different in some ways. Cardiac muscle cells, or myocytes, are shorter, they're branched, and they connect end to end via specialized junctions called intercalated discs.
Intercalated discs, what's special about them?
They contain two important things. Desmosomes, which act like rivets, physically holding the cells together against the force of contraction, and gap junctions.
Gap junctions. Those allow direct communication between cells.
Precisely. They're little channels that let ions and electrical signals pass directly from one cell to the next. This means the entire heart muscle acts like a single coordinated unit, an electrical and mechanical syncytium.
So the signal spreads easily. And unlike skeletal muscle needing a nerve signal for every contraction.
Cardiac muscle has its own intrinsic rhythm. It's initiated by specialized pacemaker cells in the sinoatrial SA node. They spontaneously generate action potentials, which then spread rapidly throughout the heart via those gap junctions, triggering a coordinated beat.
Now, you said all muscle needs calcium, but there's a critical difference in the heart regarding where the calcium comes from.
Yes, this is a huge point. Cardiac muscle contraction has an absolute requirement for C2 plus entry through L-type C2 plus channels from outside the cell during the action potential plateau. Skeletal muscle doesn't strictly need that external calcium influx, but cardiac muscle does.
So that initial influx is essential.
It's essential, and it acts as a trigger. This relatively small amount of calcium entering from outside then triggers the release of a much larger amount of calcium from the SR. This is called calcium-induced calcium release, CICR.
Ah, CICR. Calcium triggers more calcium.
Exactly. The calcium entering through the L-type channel binds to and opens the ryanodine receptors on the nearby SR, releasing the stored calcium. It's like a spark igniting a bigger fire. Each L-type channel seems to control a small cluster of IRs.
So the overall contraction strength depends on how much calcium comes in initially.
Largely, yes. And how much is subsequently released from the SR. The cross-bridge cycling mechanism itself is pretty similar to skeletal muscle, but the regulation has key differences.
Like what?
Well, for one, cardiac troponin C only has one active calcium binding site that regulates contraction, unlike the two in skeletal muscle T and C. Also, cardiac muscle has an important regulatory protein associated with the SERCA pump called phospholamban.
Phospholamban. What does it do?
In its baseline state, phospholamban inhibits the SERCA pump, slowing down calcium reuptake into the SR. But when hormones like epinephrine and adrenaline bind to receptors on the heart cell, they activate an enzyme, PKA, which phosphorylates phospholamban.
And phosphorylation changes its function.
Yes, phosphorylated phospholamban stops inhibiting SERCA. This allows the SERCA pump to work faster, pumping calcium back into the SR more quickly.
Which means the heart muscle relaxes faster.
Exactly. Fast relaxation is crucial for allowing the heart to fill properly between beats, especially when the heart rate increases, like during exercise.
That's neat. So how does the heart increase its force? Skeletal muscle uses tetanus and recruits more fibers, but the heart can't do that, right? It needs a single twitch per beat.
Correct. The heart can't use frequency summation or recruit more cells. All the cells contract with each beat. Instead, the heart regulates its force by modulating the strength of each individual twitch.
How does it do that?
Primarily by controlling the amount of calcium available to the contractile proteins during that twitch. The sympathetic nervous system, releasing norepinephrine, is a key player.
Like with phospholamban.
Yes, norepinephrine activates PKA, which does two main things to increase force. First, it phosphorylates the L-type calcium channels, causing them to stay open longer and let more calcium enter the cell during the action potential. More trigger calcium means more CICR, more cytosolic calcium, stronger contraction.
Okay, more calcium influx. What's the second thing?
PKA also phosphorylates proteins within the contractile machinery itself, increasing the sensitivity of the myofilaments to calcium. This means you get more force generated for the same amount of calcium.
Wow, so it gets more calcium and becomes more sensitive to it. Double whammy for increasing force.
Pretty much. This allows the heart to significantly increase its output when needed, like when you're running for a bus.
Smooth muscle.
Huh?
The versatile internal architect.
All right, let's move to our third type, smooth muscle, found in blood vessels, airways, gut, uterus, lots of places. How is its control different?
Very different control. It's regulated by the autonomic nervous system, sympathetic and parasympathetic, not the somatic system like skeletal muscle. And the nerve endings don't form tight neuromuscular junctions. Instead, autonomic nerves have these swellings called varicosities along their length that release neurotransmitters diffusely near the smooth muscle cells.
So it's less like a direct phone line and more like a sprinkler system.
That's a good analogy. And the receptors for these neurotransmitters are often spread widely across the muscle cell surface. Smooth muscle organization also varies.
How so?
Some smooth muscle is multi-unit. Think of the iris in your eye. Each cell gets direct nerve input and contracts fairly independently, allowing for very fine control. There are a few gap junctions connecting these cells.
Okay, independent cells.
Then there's unitary smooth muscle, sometimes called visceral smooth muscle. This is common in the gut, uterus, and many blood vessels. Here, the cells are extensively connected by gap junctions.
Ah, so they act together like the heart muscle syncytium.
Exactly. An electrical signal in one cell spreads rapidly to its neighbors, causing a wave of coordinated contraction. Many tissues are actually somewhere on a spectrum between pure multi-unit and pure unitary.
What about electrical activity? Is it always action potentials like skeletal and cardiac?
Not necessarily. This is another area where smooth muscle is super diverse. Some smooth muscle cells do generate action potentials, often slower and longer lasting than skeletal muscle, because they rely more on slower voltage-gated calcium channels for the upstroke, not fast sodium channels.
Longer action potentials, does that mean longer contractions?
It can contribute, yes. Some smooth muscles have action potentials with a prolonged plateau phase, allowing sustained calcium entry and thus sustained contraction. But crucially, many smooth muscle cells can contract without generating any action potentials at all.
No action potentials.
They can respond to neurotransmitters or hormones with just small, localized changes in membrane potential, graded potentials, that are enough to open some calcium channels. Some even contract due to chemical signals that release calcium from internal stores without any change in membrane voltage. This is called pharmacomechanical coupling.
Pharmacomechanical coupling. So drugs or hormones cause contraction directly, bypassing electrical signals.
Right. And some smooth muscle cells even exhibit spontaneous electrical activity, like pacemaker potentials that drive rhythmic contractions, or slow waves, which are oscillating changes in membrane potential that can trigger contractions or action potentials. It's incredibly varied.
So getting calcium to the cytoplasm is still key, but the ways it gets there are more diverse in smooth muscle.
Definitely. 3 main routes we can think about. 1, calcium entry through voltage-gated channels, like L-type channels, opened by depolarization, like an action potential or graded potential.
Okay, standard voltage gating.
2. Calcium release from the SI-CICR can happen, but a really important mechanism in smooth muscle is IP3-mediated calcium release.
IP3.
Yeah, when certain hormones or neurotransmitters bind to receptors on the cell surface, they trigger an internal signaling pathway that produces a molecule called IP3. IP3 then binds to specific receptors on the SR membrane, which are essentially ligand-gated calcium channels, causing calcium to be released from the SR stores.
So a chemical signal from outside leads to internal calcium release independent of voltage changes.
Exactly, that's part of pharmacomechanical coupling. And pathway 3, calcium entry through voltage-independent channels in the cell membrane. A key type here are store-operated calcium channels, SOCs.
Store-operated.
Yeah, these channels open up when the calcium stores inside the SR become depleted. It's like a signal saying, hey, the tank is low, let some calcium in from outside to refill. This helps maintain calcium levels for sustained contractions or SR refilling.
Wow. Multiple layers of control over calcium. Now, once calcium is elevated, how does smooth muscle actually contract? It doesn't have troponin in the same way, right? And it looks smooth, not striated.
Correct. It lacks the highly organized sarcomeres, so no striations. And the regulation of cross-bridge cycling is completely different. It doesn't involve troponin and tropomyosin directly blocking actin sites. Instead, the regulation happens by controlling the myosin itself.
Controlling the myosin. How?
When intracellular calcium rises in smooth muscle, it binds to a different calcium-binding protein called calmodulin, CAM.
Calmodulin, not troponin C.
Right. The calcium calmodulin complex then activates an enzyme called myosin light chain kinase, MLCK.
Okay, CAM activates MLCK.
MLCK then does exactly what its name suggests. It adds a phosphate group, phosphorylates a specific part of the myosin molecule called the regulatory light chain, which is near the myosin head.
Phosphorylates the myosin light chain. And that is the trigger.
Yes. That phosphorylation is the key step. It changes the conformation of the myosin head, dramatically increasing its AT-PACE activity and allowing it to interact with actin and go through the cross-bridge cycle. Without that phosphorylation, smooth muscle myosin is essentially off.
So, unlike skeletal muscle, where myosin is always ready and calcium just removes the blocker, here, calcium has to activate a kinase to turn the myosin on.
Precisely. It's a biochemical switch, not just a physical block being removed. This phosphorylation step is relatively slow, which is one reason why smooth muscle contraction develops much more slowly than skeletal muscle contraction.
That makes sense. How does it relax then? You need to undo that phosphorylation.
Exactly. Relaxation requires dephosphorylation of the myosin light chain. This is done primarily by another enzyme called myosin light chain phosphatase, MLCP, which removes the phosphate group.
So it's a balance between MLCK activity, turning contraction on, and MLCP activity, turning it off.
Absolutely. And the cell can regulate the activity of both these enzymes. This means smooth muscle force isn't just determined by calcium levels, it's also determined by the relative activities of MLCK and MLCP. You can even get situations, calcium sensitization, where force increases without a further rise in calcium just by inhibiting MLCP.
Fascinating. And what about that latch state?
Ah, the latch state. This is a really cool energy-saving feature of some smooth muscles. Once contraction is established and myosin is phosphorylated, even if calcium levels drop slightly and MLCK activity decreases, some myosin heads can remain attached to actin in a force-producing state for prolonged periods without cycling rapidly and consuming much ATP.
So they sort of latch on.
Kind of. It allows smooth muscle, like in blood vessel walls, to maintain tone or tension for very long durations without fatiguing and without burning huge amounts of energy. Super efficient for sustained functions.
Muscle diversity built for purpose.
Okay, we've covered the three main types: skeletal, cardiac, smooth, each unique.
Totally unique in their structure, how excitation leads to contraction, how they're regulated. But if we connect this to the bigger picture, the diversity doesn't stop there. Even within each category, there's incredible variation.
Yeah, like with skeletal muscle fibers.
Exactly. We broadly classify them into slow twitch, type 1 fibers, and fast twitch, type BIA and IB fibers. Type I fibers are your endurance fibers. They use oxygen, oxidative metabolism, have lots of mitochondria and myoglobin, making them look red, contract relatively slowly, but are very resistant to fatigue. Great for posture.
And the fast twitch.
Type 2 fibers contract much faster. Type IA are highly glycolytic, relying less on oxygen, have fewer mitochondria, look whiter, and fatigue quickly. They're built for short bursts of power. Type IA are kind of intermediate. This difference comes down to expressing different versions or isoforms of key proteins like myosin heavy chains, SERCA pumps, even troponin.
Different tools for different jobs, even within skeletal muscle.
And you see similar diversity in the heart. Atrial cells have slightly different properties than ventricular cells or the cells of the conducting system, again due to different protein isoforms.
And smooth muscle seems like the most diverse of all.
By far. Smooth muscle properties vary tremendously depending on the organ. Think about the smooth muscle in your airways versus your bladder versus a small artery. They respond differently to different signals, contract with different speeds and durations. And smooth muscle can even change its properties over time, adapting to conditions like the uterus during pregnancy.
How does it achieve such diversity?
Through expressing a huge variety of different protein isoforms, different types of receptors for neurotransmitters and hormones, different ion channels, it gives smooth muscle an incredible toolkit to respond specifically to local needs and signals. For instance, low oxygen causes smooth muscle in systemic arteries to relax, to increase blood flow, but causes smooth muscle in pulmonary arteries to contract.
Wow. Opposing effects based on location.
It's all about specialization for function.
So what does this all mean? We've journeyed from the whole muscle down to the intricate dance of actin and myosin, powered by ATP and triggered by calcium, but regulated in subtly and sometimes dramatically different ways in skeletal, cardiac, and smooth muscle.
And we've seen how this fundamental machinery is adapted and diversified to meet the specific needs of every part of the body.
It truly highlights the body's incredible adaptability and specificity, all built on these core physiological principles, but executed with remarkable molecular precision.
And understanding these principles, this detail, isn't just an academic exercise. It's fundamental. It's the language we use to understand health and disease, to develop diagnostics, and to design effective treatments. It forms the bedrock of clinical reasoning for so many conditions.
Absolutely. So as you go about your day, maybe think about this incredible machinery inside you. Consider how these precise molecular mechanisms power every move you make, keep your heart beating, regulate your blood pressure, and how glitches in this machinery are at the heart of so many diseases. What new questions does digging into this level of detail spark for you about how our bodies work or how they sometimes fail?
It's complex stuff, no doubt.
It is, but remember, you're definitely capable of mastering this. You're part of the deep dive family, breaking down these intricate systems piece by piece. Keep digging.